Hello everyone, and a gracious welcome. I'm much delighted to meet you all again today for the fifth session of this international virtual conference on frontiers in biological research, which is organized by the Department of Botany in St. Joseph's College, Tiruchirappalli, Tamilnadu, in association with the Biomics team. For the fifth session of this international virtual conference, we have another great personality, Dr. Pushpanathan Muthuirulan, who is currently working as a research associate in the Department of Human Evolutionary Biology at Harvard University, USA, is with us today, and he will be delivering his talk in this session on the topic OMICS approaches to study human diseases. On behalf of the Biomics and St. Joseph's College, Tiruchirappalli, I extend my warm welcome to you, Dr. Pushpanathan. Today, our speaker, Dr. Pushpanathan, will elaborate about omics technologies which have revolutionized the medical research and have a broad range of applications that primarily involve mining of genes, that is genomics, mRNAs, that is transcriptomics, proteins, that is proteomics, and metabolites, that is metabolomics. The routine implementation of such high-throughput omics technologies in medical research has made significant achievements, which led to development of tremendous genetic and molecular approaches that help us to gain deeper knowledge and understanding of diverse range of human diseases. Today, we will provides an overview of such technologies such as genomics, transcriptomics, proteomics, and metabolomics, and focus on methods for their integration across multiple omics layers to understand the flow of information that underlies diseases. Before stepping into the key event of this lecture, may I now invite one of our organizing secretaries, Dr. Kilimas Rajan, the Associate Professor of Zoology in the Department of Botany from St. Joseph's, to introduce our today's speaker, Dr. Pushpanathan Muthuirulan, to our audience.
Thank you, Partha Sarthi, the moderator and the chairs of the Biomics, for this wonderful opportunity. A pleasant good evening to everyone who spare your valuable time with us for attending the fifth day of the International Virtual Conference on Frontiers in Biological Research, organized by the Department of Botany, St. Joseph's College, Tiruchirappalli, in association with the Biomics Bangalore. At this juncture, I take this opportunity to thank and welcome our speaker on behalf of our college Jesuit management, Head Department of Botany, Dean School of Biological Sciences, teaching and non-teaching fraternity, and the students of botany departments, and also the scientists, research scholars, and students, and all the participants from our college and the other institutions. On behalf of all, I once again welcome you, sir. Moreover, it is my great honor and privilege to introduce our speaker, Dr. Pushpanathan, now currently working as a research associate in the Harvard University, Cambridge, USA. The overarching goal of this research is to use functional genomics approaches to understand the genetic and developmental basis of stature variation in skeletal disorders, that is the developmental dystrophys of hip and osteoarthritis in humans. For his postdoctoral work at NICHD, National Institute of Health, he developed a novel receptor-based GRASP method using CRISPR-Cas9 and a high-resolution microscopy to map neurotransmitters receptors to brain, the neural circuit that involves visual motion processing in Drosophila. For his PhD at the Department of Genetics, Madurai Kamaraj University, he had discovered a novel antifungal peptide, MMGP1, from marine metagenomes and characterized its distinct antifungal mechanism with an ultimate goal to combat opportunistic human fungal infections such as the candidiasis as well as aspergillosis, that is aspergillosis. Dr. Pushpanathan is expert in omics technologies, genetics engineering, antimicrobial drug discovery, protein engineering and mapping, neural circuits, developmental and evolutionary genetics, and also exposure to a diverse mixture of academic, industrial research experience and a diverse research group during his graduation and post-graduational research, and have allowed him to interact effectively with the scientists across various disciplines. In his current research, he tries to integrate epigenomic, genomics, transcriptomics, and targeted genetics using CRISPR to identify the target which could be translated to the clinic. And he also have a huge interest in translating his research finding to the clinic, which helps the saving billions of people lives who suffer from the skeletal diseases. He is the recipient of several awards, such as Young Researchers Award in Microbiology by Venus International Research Foundation India, Fellow Award of Research Excellence from NIH USA, 3 Minutes Talk Science Communications Award NIH USA, Junior Research Fellowship Award Lady Tata Memorial Trust India, and Meritorious Fellowship Award UGC India. So thus, we have an eminent research scientist with us. So we are very proud and happy to have you as a research person for today's section. Thank you, sir. Thank you, one and all. So now over to Professor.
So thank you, sir. Thanks for a nice introduction. So should I start sharing my screen?
Yeah, you can start your presentation, sir.
Sure. So can you able to see my screen?
Yes, it's all right, sir.
Yeah. So thank you so much. First of all, I would like to thank all the organizers for inviting me for this wonderful session. So I'm very happy to be here, and it's a pleasure to give talk over this conference. Thank you so much. So today I'm going to talk about the important thing that people ignore in most of time is like the OMICS approaches. So at the end of this talk, you will get to know why the OMICS approach is important to understand the human diseases. So I'll be briefly going through all the approaches that you can use for understanding human diseases. So if you have any questions, like I'll be just focusing on some of the techniques. If you have any more the questions, you feel free to reach out to me this email address. This is my email. So you can contact me anytime if you have any question about my presentation. So the topic of my presentation is about the OMICS approaches to understand human diseases. So as you know, the human body contains trillions of cells. Like the cells, it looks like this, and the cells has the chromosomes, DNA, and the DNA has genes, and the genes contain the instruction that makes the proteins. And the proteins can act alone or in complex, and it perform many cellular functions. So this is the basic, the cellular process that is happening within the cells. And if you look at the central dogma in biology, so we have the DNA. So once it transcribed, we'll get the mRNA, and the mRNA translated to protein. This is the central dogma in biology. And this is very important for the cellular process because this is what happening within the cells. The genotype that determines the phenotype. Mainly your DNA that determines the phenotype of the individual. For instance, if your DNA is not good, for instance, if you have any mutation in the DNA, that is your genotype that leads to the disease phenotype. For instance, if you are exposed to any of the carcinogens, so that may lead to a disease phenotype such as cancer. So this is very important process. This flow of information is very important for the cell to maintain its homeostasis and to function in a proper way. So this is the main important thing. So the main talk of the most of my talk, I'll be presenting the concept, like how the genotype can be establish the phenotype, like how genotype is important to maintain the phenotype. So this is the conceptual model of human disease. So you may ask the question, why some people used to get disease and some people are healthy? So that is mainly because of the environmental exposure. It depends on the environmental factors. For instance, if you take some of people used to smoke, So the smokers, what happens? Like if it is a long-term smoker, so what's happening there that may affect any of the constituents within the cell. It may affect the genome, epigenome, transcriptome, proteome, or metabolome. So in case, let's assume if you are a long-term smoker, that affects the epigenome, like it affects the modification of the genome. So that may cause to the lung cancer. That is one of the disease phenotype. This is just one example I'm just explaining. Similarly, the other environmental factors, such as let's assume if you are exposed to the ultraviolet radiation. So if you are exposed to the UV radiation, so that may create mutations in the genome. So that may lead to cancer. So the environmental exposure is one of the important things that is responsible for the disease phenotypes. And also, if you look at the current research, the most of the research are focused at the individual level. For instance, so some of the researchers are mainly focusing only on the genome. They want to know what is happening at the genome level. And the other researchers might be focusing only on the epigenome. Epigenome is a modification which happens in the genome. And some labs are focusing on transcriptome, proteome, and metabolome. The problem right now here is, so there is no integration between these things because the disease can happen at any stage. The disease can happen at the level of genome, epigenome, transcriptome, proteome, or metabolome. So it's very important to integrate all these things to understand the complete mechanism or the pathophysiology of the diseases. So currently we are lacking this integration pipeline so that we don't have the complete picture of what causes the disease. We don't have the complete details of how this disease is caused. So whatever the treatment we have right now is not applicable to treat the disease completely. So it's incomplete. So the disease can happen at any stage. It can happen at the newborn, for instance. These are some of the disease in the newborns, like at the neonatal stages, like the low birth weight, the congenital abnormalities, and also at the toddler and the child. You can see there is the respiratory diseases and also some of neurodevelopmental problems. And also the adults may face some of disease like the cardiovascular diseases, cancer, the overweight, that's obesity. So the reason for all these things are, it's an environmental exposure and also the lifestyle modification. Like some people used to drink, some people used to smoke, and some might be exposed to the carcinogen, especially during their workplaces, all these things. So this may have a longer impact on the human health, and that leads to the disease condition. So it's not something like the disease happen at one particular stage. It happen at the different stages of the human development, especially at neonatal, toddler, child, adolescent, and adult stages. So it's very important to understand the disease mechanism at all these stages. So let me explain the term omics. So the omics, it refers to the field of study in the biology. This is something like the field that ends with omics. For instance, if you take genomics, it ends with omics. So this comes under the omics platform, and also the transcriptomics, metabolomics, and proteomics. So I'll explain each of these things in more detail in the next slide. So the main aim of the omics is to collectively characterize and quantify the pool of biological molecules. As I mentioned, the biological molecules can be genome, transcriptome, proteome, or metabolome. So the main aim of the omics is to collectively characterize and quantify the pool of biological molecules that translate into structure, function, and dynamics of the organism. So this is the main aim of the omics approaches. So the scope of omics is to determine the biomarkers for disease. So this is one of the application of the omics. So you can determine the biomarker for disease. So if you know the biomarker, so it will be easy for the diagnosis. Like you can use the biomarker for disease diagnosis. And the omics also differentiate the biological pathways or processes between disease and the control groups. And it also offers greater understanding of flow of information from original cause of the disease. So this is what we are lacking right now. We don't know what is the original cause of the diseases. It may be a developmental, environmental, or genetic cause. So using omic approach, we can better understand the flow of information from the original cause of disease to the functional consequences or the relevant interaction. So the main important application of the omics it's to practice the precision medicine, which I'll be focusing in the rest of my talk. So as I mentioned, so this is the individual constituents that is happening within the cells. Like we have a genome, epigenome, transcriptome, proteome, and metabolome. So I'll be going through the each field, individual things, and also I'll be describing the approaches and the application of each omics technologies. So first I'll focus on the genomics. So the genomics, I think most of you are aware of the term genomics. Genomics is a study of genome structure, function, evolution, and mapping. So in genomics, people used to study genes, the gene regulatory elements such as promoter, enhancers, and also the genetic changes that are happening within the genome. For instance, there may be changes in the single nucleotide polymorphism, or there may be insertion, deletion. So all these things constitute the field genomics. And there are several approaches used for understanding the genome function and structure. So these are some of the approaches you can use for understanding the genome structure and function. For instance, the whole genome sequencing, genome-wide association studies, genotyping arrays, exome sequencing, genome editing with CRISPR. So here I listed the genomic approaches, whatever I mentioned before, the whole genome sequencing. You can use the whole genome sequencing to detect the single nucleotide variants or insertions or deletions in the genome or copy number changes or the structural variants in the genome. This is one of the approach you can use to look at the genetic changes especially. And GWAS is it's a genome-wide association study, and this is an important, like now you can see a lot of the disease they are performing the genome-wide association study. So these GWAS studies, it associates the specific genetic variation with particular disease. So what exactly people used to do here, they will sequence the genome of the healthy and the patient, and then they'll compare the two genomes. If you compare the genome of the healthy and the patient genome, so you would expect to see a difference. So that difference would be the causative reason for the particular diseases. So using genome-wide association study, you can understand the genetic changes associated with the disease. The next approach is the genotyping arrays. So genotyping array also explores the genetic variants such as single nucleotide polymorphism, like small changes happening within the genome. Or you can also look at the larger structural changes in the DNA. The next approach, genomic approach, is the exome sequencing. The exome sequencing, if you look at the human genome, only the 3% of the genome that codes for protein. So exome, so what people used to do, the main advantage of exome sequencing is, because if you sequence the whole genome, let's assume if you are sequencing this whole genome, it is too expensive. The reason is like you are sequencing the complete genome and you need to spend a lot of money in sequencing. But the exome sequencing is like you are just focusing only on the coding sequence. Coding sequence in the sense you are just focusing only on the regions which are coding for protein. So here the exome sequence is less expensive, but it provides more information about the genes or the genetic cause of any disease. And the next approach, genomic approach, is the CRISPR. As you know that the CRISPR received the Nobel Prize recently. The CRISPR technique, suppose if you know any of the targets or genes that is responsible for disease, so you can use this CRISPR approach to functionally validate those genes. Let's assume you identify some genes that is responsible for diabetes. You want to further validate those genes. So what you can do, you can just you can derive a cell line and you can use a CRISPR experiment. You can delete the genes from the genome and look at the function of those genes. So the CRISPR experiment is mainly for the functional genomics approaches. So you can validate the function of any genes. Not only genes, any regions in the genome. So the second approach I'm going to focus here is the epigenomics. So now we know what is genomics. Just the genomics is a study of the genome structure and function. The second thing is epigenomics. To look at the epigenomics is nothing but you are studying the chemical modification of genome. So you can see at the genome level, there are a lot of modifications are happening within the genome. For instance, one of modification is like the DNA methylation and acetylation, histone acetylation. So what happened with this modification is like once these groups are added to the DNA, that may activate or repress the particular regions in the DNA. So that affects the gene expression. So this epigenetic modification is really important for the DNA to be expressed or repressed within the cell. So So the understanding epigenetic modification is really important because this also might be one of the reasons that drives the disease phenotype. So as I mentioned, in epigenomics, we are studying the chemical modification of genomes, such as DNA methylation, the chromatin modification, and chromatin topology. So these are some of the approaches that you can use for understanding the epigenome. So here you can all see the approaches here. One approach is the bisulfate sequencing. This is one of the high throughput sequencing approach. You can look at the DNA methylation patterns. So using the sequencing approach, you can see where the DNA methylation is happening within the genome. And the second approach is the attack sequencing. Because if you look at the human genome, not all regions in the genomes are functionally active. So some of the regions are active, some of the regions are not active. Using the attack sequencing, you can detect the accessible chromatin in the genome. Accessible chromatin in the sense, if you use this attack sequencing approach, you can identify the regions which are functional in the genome. So if you know which region is functional in the genome, you know which region is responsible for the cellular processes. For instance, if you have attack sequencing results from the healthy individual and if you have the attack sequencing results from the patient, if you compare both results, you can see might be like the patients might have some of the regions which are not active in the genome. Maybe might be the causal mechanism for the disease, so you can develop some therapeutics against that particular region. Similarly, the another approach is the DNA-seq, where you can determine the chromatin accessibility, similar to the attack sequencing. Both are same, but the enzymes that used for the sequencing approach is different. And another approach is epigenomic approach is the chip sequencing. The chip sequencing where you can determine the DNA-protein interaction. And also, now people started using these techniques. This is known as the chromosome conformation capture. This is also known as 3C, 4C, 5C, high C. So this is the different version of the technique. So what you can really do with this technique is you can map the spatial chromatin organization. So similar to protein, as you may know, that protein is folded in the three-dimensional structure. Similarly, the human genome is also folded in the three-dimensional conformation. So if you look at the 3D conformation of the genome, that would provide an important insight into the disease mechanism. So using this chromosome conformation capture technology, like you can use any of this technology, so you can map the spatial chromatin organization. If you know the spatial chromatin organization within the genome, you can really get more details about what makes changes in the genome that causes the human diseases. So this is the emerging technologies now people start using to understand the epigenetic changes in the human genome. So the next thing I want to focus is the transcriptomics. As I mentioned, we have a genome. If you study the genome, that's the genomics. And when the DNA is transcribed, you'll get the RNA. If you study the RNA transcript produced by the genome, that is called the transcriptomics. So the RNA could be of anything. It could be of messenger RNA, ribosomal RNA, or the tRNA. So the transcriptomic approaches targets the different types of RNA. You can sequence the mRNA, tRNA, ribosomal RNA, and you can relate them with any of the disease. For instance, we can study the coding RNA, which is the mRNA, and non-coding RNA species, such as the long-coding RNA. This is the long non-coding RNA and microRNA and small RNA. So now if you look at the recent publication, you can see the researchers keep on reporting a lot of microRNAs and small RNAs that have been associated with cancer, diabetes, cardiovascular disease, and all this. So it's very important to study all these RNA in relevance to the disease phenotype. So these are some of the transcriptomic approaches you can use to understand the RNA transcript produced by the genome. So one such method is the RNA sequencing. I think most people are familiar with this technology. So using RNA sequencing technology, you can study the regulation of gene expression, how the gene is expressed. And using microarray, again, you can look at the regulation of gene expression. But people prefer RNA sequencing than the microarray because the RNA sequencing is less expensive and also you can perform at a very high throughput. Even microarray you can perform at high throughput, but you need the specific probes to look at the gene expression. But in case of RNA sequencing, So no such probes are needed. You can directly sequence the RNA to get more information about the transcriptome. And this is the latest technology, which is called the iClip, which is called the cross-linking immunoprecipitation. Using this approach, you can identify the protein-RNA interaction. So these are some of the approaches you can use to understand the transcriptome. So let's move on to the proteomics. So the proteomics is something like you have your DNA, if it is transcribed, you have the mRNA or tRNA, ribosomal RNA, whatever it is. If this gets translated, if the mRNA gets translated, you'll end up getting a protein. So the proteomics is nothing but you are studying the proteins which is encoded by the genome. So there are a lot of approaches available to study the proteome. So here I have listed all the proteomics approaches that you can use to study the proteome. For instance, you can use the label-free or metabolic labeling or chemical labeling approaches to study the quantitative proteomics. So quantitative proteomics, what exactly you are doing here is you are quantifying the proteome. For instance, you have the healthy individual and the patient. If you do the quantitative proteomics between the healthy individual and the patient, you can definitely see some differences because the patients might have a different level of proteomes. So you can relate those results with the disease phenotype. So these are some of the approaches you can use. And these are another approaches like the affinity purification mass spectrometry, the cross-linking mass spectrometry, and ion mobility mass spectrometry. These are some of the approaches you can use for the interaction proteomics. The interaction proteomics in the sense, so one protein can interact with other proteins within the cells to regulate the cellular function. So to understand the protein-protein interaction, you can make use of these approaches. Similarly, you can also for the organelle proteomics, the cell have a different compartment like the cytoplasm, nucleus, mitochondria. So using the organelle proteomics, you can study the proteome which is specifically present within the organelles. For instance, if you want to study the mitochondrial proteins, so you can make use of these approaches to understand the protein in specific compartments such as the cytoplasm, nucleus. And also another approach is immobilized metal affinity chromatography and metal oxide affinity chromatography. It's a chromatographic technique which can be used for the phosphoproteomics. Because if you look at the protein has several modifications like phosphate the protein phosphorylation. So far we talk about the genomics and the approaches and application, epigenomics, transcriptomics, and proteomics. So I just listed the approaches, but I didn't get into more details. Maybe if you have any question, you can reach out to me later. So the last thing I want to talk about is the metabolomics. So you can see the DNA transcribed to RNA and RNA translated to protein. And the last thing is the metabolites. The metabolites are generated from the metabolic reactions catalyzed by the enzymes. So it's something like this. So the metabolites can be anything like a glucose inside the body or the lipids, the glycans, carbohydrates, or amino acids. So these are the metabolites. So it is also very important to look at the metabolites during the disease condition. For instance, if you take a person who is diabetes, so you can look at the metabolites. If you look at the blood glucose level in the diabetes patient, so that metabolites provide some information. If the glucose level is more, it says the person is having diabetes. So measuring the metabolites is very important, and it provides insight into the disease progression, whether it is an indicative of any particular disease. For instance, as I mentioned, if you look at the glucose, if it's high, if it is beyond the normal range, it says the person is diabetic. So measuring the metabolites is one of the important things in understanding the disease mechanism. So the metabolomics is nothing but you are studying the metabolites generated from the metabolic reaction, which is catalyzed by the enzyme. So now you can look at these things. Like you have the DNA. If you study DNA, that's the genomics. And if you study the RNA, that is transcriptomics. And if you look at a set of protein within the cell, that is the proteomics. And if you study the metabolites, it's all metabolomics. So the problem here is, as I mentioned before, so the researchers start using one particular approach. For instance, they use only the genomic approaches to might be used one let's take in particular disease if the transcriptome get affected and that leads to any disease phenotype. But if you are focusing only on the genomics, it means you are missing some information and you are not having the complete picture of what is exactly happening within the cell that leads to the disease mechanism. So this is, as I mentioned, this is one of the approaches that you can use to study the metabolites. It's a stable isotope labeling followed by the mass spectrometry. So the next one thing I need to focus on is the microbiomics. So far I showed it all. You can see there is a flow of information. The genomics, epigenomics, it is a continuous flow of information. The next thing is the microbiomics. So the microbiomics is studying the microbial population within the human. For instance, you can see the human gut microbiome has been reported to be involved in many of the diseases such as cancer, diabetes, cardiovascular disease. So it's very important to study the human microbiome. That is one of the OMICS approaches. So let's take the COVID-19. So you might be aware that people having these risk factors are more susceptible to the COVID-19 infection. For instance, the people with hypertension, cholesterol, obesity, and excess use of antibiotics. Some people, like because of the COVID-19 restriction, the dietary modification, like people start eating the home food and they may have a different lifestyle, and people are more stressed during COVID-19, and diabetes, infection. So all these risk factors, so people with all these risk factors have more susceptible to COVID-19 infection. So this might be one of the reasons. For instance, if you take a healthy human, this is the microbiome. Let's take this is a human microbiome. So this is a healthy microbiome. So if people have any of these risk factors, for instance, hypertension and cholesterol, what exactly happening here is that disrupted your microbiome. For instance, this we call as a microbial dysbiosis. So these risk factors can modulate your microbiome. So the main function of the human microbiome is it maintains the homeostasis within your body, and it is responsible for maintaining the proper immune response. So if your microbiome got disrupted, so in this case, what's happening is your immune response is not really good as the healthy person. So this might be one of the reasons. Like if you have any of the risk factors that will affect your human microbiome. So if you got any of infection, not only the COVID-19, maybe of any of other infection like a bacterial infections or any other infection, so that might be more severe compared to the healthy individual. So it's important to maintain a healthy microbiome. So this is one of the omics approaches that you can use to understand the disease mechanism. As I mentioned, the omics areas involve the quantitative analysis of all the constituents of systems. So let's take the genomics, transcriptomics, proteomics, and metabolomics. So the omics approach is take into consideration of all the constituents, not a single entity. It's not only focusing on genomics. It's not only focusing on proteomics or transcriptomics, proteomics, or metabolomics. The omics, it take into consideration of all the constituents in the cells. For instance, genome, transcriptome, proteome, and metabolome. So how you can really integrate all these things? It's mainly by making use of the system biology. By using the bioinformatic approaches, you can really integrate all these systems together. You can really integrate all these data. So as I mentioned, there are different approaches to understand genome, transcriptome, proteome, and metabolome. Once you have the results from all the approaches, you can really use the system biology to integrate all these results. So that's the main focus of the omics, is to integrate all the constituents within the system. So this is the medicine today. This is the medicine we have currently. For instance, this is our expectation. Let's assume if people have a cardiovascular disease, what our expectation is, we want one medicine that should treat the cardiovascular disease in all patients. So this we call as one size fits all. So this is our expectation. We want one medicine. You just want to give one medicine to all the patients with cardiovascular disease. It has to cure the disease. But in reality, this is what happening in reality. So the one size doesn't fit all. So to be in the real world, if you take a medicine, for instance, if you take a cancer, if you give drug to 100 cancer patients, it's not going to treat 100 cancer patients. Some persons are responsive to the drug and some people may not responsive to the drug. The reason is because the disease, we don't have the complete understanding of the disease mechanism. We don't know what exactly is happening within the cell, and we don't have the complete information about the disease mechanism. So without the complete knowledge, if you provide any of the drug or vaccine or whatever to the patient, it's not going to be 100% effective. So this is our expectation, but this is what the reality is about. So the main goal of the OMICS is the precision medicine. So this is the proposed model of the omics tech approaches. Like you can use an omic approaches to practice the precision medicine. So I'll just briefly explain how you can use the omics approaches to use for the precision medicine. So let's take a patient. It may be of any patient. The first thing is you have to create an omics profile. So we discussed about all these approaches. And we also know the application of each approaches, like how you can understand the genomics and also what are the application of the genomic approaches. The first thing is you need to create an omics profile from a patient. Like you have to create a profile for genomics, epigenomics, transcriptomics, proteomics, metabolomics, and microbiomics. So once you have the omics profile, the next thing you want to do is you have to integrate all the data together. So you can use the system biology, bioinformatics, and biostatistics to integrate all the data, and you need to analyze and interpret the results. Once you integrate all the data, and if you analyze and interpret the results, you will end up getting a complex disease network. So in this case, what you are doing, you are considering all the constituents in the system. You are not just focusing only on genomics, epigenomics, transcriptomics, or proteomics. Here you will be taking into consideration of all the system. You are just looking at what is exactly happening within the cell at the different layers, like different layers of biological molecules. For instance, you are seeing what is happening at DNA, what is happening at epigenome, what is happening at RNA, what's happening at proteome, metabolome. Even you are still considering the microbiome, what is happening within the microbial community in the patients. So if you created a omics profile from an individual patient, and if you integrate the data, and if you analyze and interpret the results, you would understand the complex disease mechanism. So after analysis, you will end up getting a disease signature. Disease signature in the sense, it will give you a clear picture of what is the disease mechanism. So what causes this disease? So using this complex disease network, then you can develop a clinical care. So if you use this approach, you can use for the personalized therapy. For instance, you can develop a drug for individualized patient. For example, if you have 100 cancer patients, let's say, if you develop one drug for 100 patients, definitely it's not going to treat the cancer completely. So not all 100 persons are going to get cured out of one medicine. So in each case, what you have to do, you have to create a complete omics profile, and you have to integrate the results and come up with the disease mechanism for each individual person. And you have to develop a therapy for therapy based on the patient's genetic makeup. So that's what we call as the precision medicine. So the precision medicine is a medical model that proposes the customization of the healthcare, which is tailored to a subgroup of patients. You are just focusing on one particular patient, not considering all the patients and trying to develop one drug. So this is the main application of the omics approach. So with that, I would like to thank you all for your patience. So if you have any questions, I'm happy to answer any of the questions you have. So any questions?
Yeah, thank you so much, sir, for your wonderful presentation that you elaborate about various omics technologies in medical researchers like and even micro all just together and a way of flow of information that underlies different diseases. Thank you so much, sir. And with your permission, I will forward some of your few questions from your conclusion of is that you have concluded that about the precision medicine when it comes to precision medicine by
Yes.
analyzing a single individual and taking the profile of a single individual and making a data, integrating the data and complex network, making a complex disease network and giving a treatment for that particular individual that is called precision medicine. Does this is applicable in this modern world? That then the next question is it affordable? One is applicable, then is affordable.
Yeah, actually, there is like already the evidences are there. People start using, for instance, if you take in the place I live here in Cambridge, Massachusetts, what they are people there is an organization they used to collect the stool samples from the healthy donors. Like we don't care about the stool samples, but here what they are doing, they are collecting the stool samples from the healthy donors. For instance, if I am a patient, let's assume I have a diabetes. So what they do is like they will completely profile my microbiome. So they already know the microbiome which is existing in the healthy population, and they know the patient microbiome. Let's take I'm a patient, and they know my microbiome. They want to find which part of microbiome I'm missing. Like they want to identify the signatures I'm missing in the microbiome. So from the healthy donors, they are trying to culture that particular microbiome, and they want to just do the transplantation. They can transplant those microbiome to the patients to cure some diseases. So this is what the real thing which is happening in Cambridge, Massachusetts. So there are healthy donors who offer the microbiome. They usually take the stool samples and they culture the microbiome and they will transplant into the patients. For instance, the patient is having a diabetes. So if the microbiome is the real cause for diabetes, so they know which microbiome is responsible, like which microbiome is missing in the patient, so that causes this disease. So you can really, like you can do the transplantation from the healthy donors to cure the disease. So this is one of the precision medicine, like you can individualize the treatment based on the patient microbiome. Like if you know the patient microbiome, you can do some kind of transplantation to cure the disease. This is for microbiome. Still, the omics approach people start using, and it is feasible because we are in the world where the next generation sequencing is possible. So whatever the technology I showed you, like the genomics, transcriptomics, this takes, it's not like a difficult thing to do. Making use of the latest high throughput sequencing technology, this is feasible. Like the only thing we are lacking right now is like the data integration. We can easily use the high throughput sequencing technologies to study genome, transcriptome, proteome, and metabolome. The only thing we are lacking right now is how to integrate the data. So we should need a strong bioinformatician. Can afford for lifelong. Like that will be more expensive than just understanding the disease mechanism and just cure the disease instantly. So it is applicable and also it's feasible to do.
Yes, sir. Then, as you talked about microbiome, the gut microbiome and different microbiomes, and analyzing a microbiome, only microbiome, it doesn't precision medicine, isn't it? As you say.
Analyzing yeah, you can just microbiomics is completely a different thing. So I showed you the different things, like I showed the flow of information from genomics to metabolomics, cardiovascular disease, diabetes. They are relating almost the microbiome with all the diseases now, to be honest, if you look at the literature. So that's the thing you want to really integrate this microbiomics into the platform. But if you really, I don't I'm not sure about like whether the microbiomics can be used as a precision medicine as a alone. But if you want to understand the complete mechanism, the reason like microbiomics is just one thing that contributes to disease. There might also be the other thing. For instance, I showed one of the slides which is related to COVID-19. If you see other risk factors in the body, for instance, hypertension and cholesterol. So these risk factors that causes the microbiome alteration. So in this case, the actual reason is not the microbiome changes that causes disease. You already have some of the risk factors in your body. In addition to this, you have the microbial imbalance. So that causes the disease phenotype, like the disease condition will be more severe. So my suggestion would be to integrate all these things, including the microbiomics with all other omics approaches to develop the precision medicine. That will be more effective than just looking at the microbiome alone.
Yeah, sure, sir. Then coming to the integration point of view, and when it comes to genomics, epigenomics, and transcriptomics, metabolomics, proteomics, and every stages has their own code or genetic codes. And how do bioinformaticians integrate all such codes? Why? Because each of these stages have their own codes, have their own messages in it. So what is the difficulties of bioinformaticians to integrate those things?
Yeah, that's a good question. That's what because I didn't include those information in my presentation. So currently we are doing the similar kind of work in our lab. We are trying to use the epigenomics. We are integrating epigenomics with the transcriptomics. Later on, our goal is to integrate it with the proteomics and metabolomics. If you take epigenomics, let's take in epigenomics, you are looking at, let's take a genomics first. In genomics, you are looking at the genetic changes. So if you use the genomic approaches, you know the genetic changes. Let's assume you know one region that get affected. For instance, there is a mutation in the genome. So using genomic approaches, what you did is like you discovered a novel mutation that you are thinking that might be responsible for disease. So now we have one layer of information from genomics. So the next level is epigenomics. If you do the epigenomic studies, so what you can do, so you know there is one mutation in that region. So if you integrate these two data, so integration is nothing but you have your data for each layer just to analyze them, just based on like you should have a pipeline to integrate them. So as I mentioned, you have a mutation in your genome. And now you have your epigenomics profile. Epigenomics profiles will give the modification in the genome. Suppose the DNA get methylated or the histone get acetylated. So this kind of information you will get from epigenomics. So now if you integrate this genomics with epigenomics, let's assume you have a healthy person in the sense who is not having that mutation. You have a patient who is having that mutation. So this you identified by the genomics approach. And also you are performing the epigenomics on both healthy and patients. So now if you analyze results, what exactly you can infer? So the patient is having a mutation. And that mutation is causing some changes in the epigenome because it's all are interconnected. It's not something like genome is separate and epigenome. So if there is any mutation, that may cause us some changes in the DNA modification, which is not same as the healthy person. Patient may have a different epigenome profile. So if you just look at these two data, you know that patient is having a mutation and they also have changes in the epigenome. Then next thing is you have to do the transcriptome. Transcriptome, you are really seeing the gene expression changes. So what you can get to know from the healthy and the patient comparison, you'll know like the patient have a different gene expression profile. So now you are adding the additional layer. If you integrate the three things like the genomics, epigenomics, and the transcriptomics, this is what currently we are doing in our lab. So what you are exactly seeing in patient is the patient have mutation. So that mutation causes a changes in the epigenetic modification. So because of that mutation, the DNA is not getting methylated. Let's assume no methylation. And if no methylation, so no gene is expressed. So you would clearly see all these differences in each layer if you integrate them. Next thing is proteomics. If you use a mass spectrometry and if you do proteomics, then that is an additional layer of information. So now you are adding up evidence to all your hypotheses. There is a mutation that affects DNA methylation, that affects the gene expression. So there is no gene expression, so there is no protein. It means you are validating each and every step that will provide a clear picture of the disease mechanism. So the integration pipeline is it's simple. Like you have to do the same analysis whatever we are doing for each approaches. You just analyze your genome, analyze your transcriptome. The final thing is you have to just look at, suppose if you know the region which is get affected, you can just focus on that region and see whether the protein get affected or not. So the integration pipeline is simple, but if you look at the whole genome level, I'm just talking about one mutation, right? Let's assume the patient is having 100 mutations. Then the complete genome profile will change, like the epigenome, transcriptome. So in that case, we really need a strong platform, like some kind of pipeline is really needed, and it should be validated first thing. We should make sure that whatever the pipeline we develop, it can be applicable. So that's the thing, but it's feasible. Whatever we are doing right now, we are just integrating genomics, epigenomics, and transcriptomics. So in our lab, and the results are more reliable. Like whatever we did so far, we do see some interesting results with it.
Yes. From your point, I could come to understand that the genomics data has been collected from a healthy individual, whereas the epigenomics data has been collected from the patients, right? From that, you compare.
So if you want to really understand the disease mechanism, you should have a data from healthy and also the patient, so that you can compare both. Suppose you need a control, right? You have a healthy person, which is who is not having the mutation, and you are having the patients who are having the mutation. So you should create an OMICS profile for both healthy and the disease patient.
So the control is obviously a genomics data, right?
The control is like each level you should have a control. Like for the genome, you should have a genomic control for healthy. So you should have a genomic information from healthy and the patient. And also you should have epigenome information from the healthy and patient. Like you have to create an individual omic profiles for the healthy and the patient. Then only you will come to know what is wrong with the
It's very specific to microRNAs and all other small RNAs. What is the reason behind that? What may be the reason behind that?
Actually, the microRNA, you can see the microRNA that doesn't encode for any of the proteins, right? If you take the messenger RNA that codes for protein, it's only coding for protein. But if you look at the microRNA, the main function of the microRNA that epigenetically control all these DNA modifications. For instance, it will add this methyl group. It can change the chromatin topology, like it will cause changes to the histone acetylation. So these are some of the epigenetic markers that cause epigenetic modification. So if you understand this microRNA, for instance, let's create an omics profile for a patient, and you identified that patient is having a genetic mutation, that they have the affected epigenome profile. And if you still look at the microRNA, and you found some different RNA was expressed in patients, like which is not expressed in the healthy individual. And you can really focus on that microRNA, and you can look at the epigenetic modifications. And this will give some more information about the mechanism. Like for instance, the patients have a new set of microRNA. So this new set of microRNA that regulates the epigenetic modification. So that in turns cause changes in the gene expression. So these are epigenetic markers like the small RNA and microRNA, and it is also developmentally at different stages of human development, different microRNAs are expressed to regulate different gene expression. So it's very important to understand all these small RNAs and microRNAs in relevance to disease.
Yeah. So can you explain the things going in a laboratory in which you are researching in specific to a disease in which you are applying these omic technologies?
Can I share one of the slides I can show you now? It's not a slide because that will have an overall scheme. I can quickly just
Sure, sir.
Because that would be easy for me to explain what. 1 second.
Yeah, sure.
So
doesn't response. Yes.
You can see.
Yeah.
Can
Yes,
you see
sir.
my slides now? So this is what currently we are doing. My current work is focused towards understanding the genetic mechanism of two skeletal disease. One is the developmental dysplasia of hip and also the knee osteoarthritis. So what we did here is we know one particular gene which is called the GDF5, which is the growth differentiation factor 5. We know this is a protein which play an important role in skeletal development. So how we identified this gene is through the transcriptome profile. We did a lot of RNA sequencing on the hip dysplasia and the knee osteoarthritis patient, and we found this gene is differentially expressed, the GDF5 gene. So then we want to focus on that one particular gene. So what we did is we make use of the genome-wide association studies. As I mentioned, it's a genomic approach. So what we did is we took the healthy genome from the a normal individual, and also we sequence the genome from the patients who are having the developmental dysplasia of hip and osteoarthritis. So by comparing the healthy and the patient genome, you would expect to see the genetic changes. Definitely, there would be some genetic changes that causes these diseases. For instance, this is the gene I said I'm focusing on my research work, and you can see all these dotted I mentioned here. These are the genetic changes. If you look at these genes, near to this genetic changes, near to this gene, what you are seeing is there are a lot of genetic changes. So how we identify these genetic changes is by sequencing the normal genome and also the genome from the patients who are having the skeletal disease. And by comparing the two genomes, you would expect to see the genetic changes. So I just plotted all these dots here, which is all the genetic changes. So now you can ask a question. There are these many genetic changes near to this gene. So which changes really affect this gene function? So here what happened? You are getting more information. You have more regions, like all these dots are there. We don't know which region is really affecting this gene function. So to understand we want to go to next level. We want to understand which region affects which genetic changes affects the gene expression. Then what we did is we used an epigenomics approach. We used an attack sequencing. So attack sequencing, as I mentioned in my presentation, using attack sequencing, you can able to look at the region which is open in the genome. Open in the sense the region which are active in the genome. So what we did is we perform attack sequencing and we integrated genomic results with the epigenomic results. So when we integrated, what we found is we found two regions are active in the genome. For instance, I highlighted in the box here and this box. So this GRO1 and R4 are two enhancers. And we found these two enhancers are active in the genome. How we found is using the epigenomic approaches. And if you integrate these two data sets, what exactly you are seeing here, in the genome, in the human genome, there are two regions which are active. And by integrating with the genomic data sets, what you are getting to know, so this is the region which are active in the genome, and this region has some genetic changes. That's why we mark this two regions. So from integrating genomic and epigenomic data sets, we refined to two particular regions. Here you can see hundreds of regions are here. We don't know which one is affecting this gene expression to cause disease. By using the attack sequencing approach, we refined to two particular regions. Now from 100, we just reduced to 2, and we know these two regions might be responsible for regulating this gene expression. Then what we did is we tried to knock out this using CRISPR technology. This is what we did. We used the cell lines and we knock out these two regions and we studied this GDF5 gene expression. So what we observed is if you knock out these two regions from the genome, that decreases the GDF5 expression. So this provides a solid evidence that these two enhancers are regulating this GDF5 expression. Then we also tested the same thing in mice. We created the knockout mice using CRISPR technology. We knock out these GRO1 enhancer and R4 enhancer in the mice separately. And again, we look at the GDF5 expression. So interestingly, what we observed in our research is if you knock out this GRO1 enhancer, that mice is getting hip dysplasia. Hip dysplasia in the sense the hip of the mice get affected. But if you knock out this R4 enhancer, that is affecting the GDF5 expression in the knee, and these mice are getting knee osteoarthritis. So this is an important finding because there are two enhancers regulating one gene expression. But the GRO1 is regulating this gene expression only in the hip region. That's why the people who is having a defect in this GRO1 enhancer, they got hip dysplasia. And this R4 enhancer is expressed in knee, and it regulates the GDF expression in the knee region. So if anybody have a mutation or any changes in this R4 enhancer, that affects this R4 enhancer expression, and that in turn affects the GDF expression. So this is two enhancer which is connected to two different diseases. And especially the skeletal disease are not simple, it's a complex disease. It has to undergo a lot of biological mechanism, and that drives the disease phenotype. So this is just, I'm showing here two things. Are what we are currently doing is we have the genomic data, we have the epigenomic data. By integrating these two, we identify two enhancers. And currently what we are doing, we also have the transcriptome data. We want to integrate the transcriptome data to see what are the other genes get affected, because there are also other variations here. Because we found these two changes are associated with this gene expression, still other changes may be associated with other gene expression. We don't know what exactly it's doing. So we are trying to integrate this with the RNA-seq and also the proteomics data sets to have a complete picture of what is happening at the disease level. So this is just one thing I just want to share, but still it is not complete. We didn't create the complete profile, but we are still on the way. Like, this is just two things I want to show. Like, we created a genomic and epigenomic profile. Even using this two profiles, we end up getting a two interesting enhancer that causes two different diseases. Similarly, if you integrate more layers like transcriptome and proteomics, you will get more targets. So you can potentially target any of the regions, and you can develop therapeutics, especially for the precision medicine. Yeah, this is just one example I'm showing. This is what the work I'm currently doing right now.
Yeah, thank you. So it's just surprising that targeting a single gene, is obviously getting resulted into two different enhancers, which is meant for two different diseases in a single gene. Is quite surprising. It's thank you so much, sir, for your presenting your research work here and clearing all our doubts. It's really a quite interesting sessions of OMICS technologies. Now, I request the panelists over here, if you have any queries, you can directly ask to the speaker.
So thank you so much for your lucid presentation, sir. So there is one question. So usually we are saying that the profile, the OMICS profile, no? The OMICS profile is applicable in normal situation, whether your patient is admitted in the hospital for treatment, whether they are switch over to this type of OMICS profile and then they are for the treatment. It is possible.
Yeah, it is possible, sir. That's what I said. Like, if the platform, the integration platform is available, now we can do all this. Like now, if you take 50 years back, I don't know, like whether people have the diagnostic method to do even for the blood test, sugar test. I don't know. People can test diabetes or not. It is not feasible 50 or 60 years back. They don't have any kits or something to test the diabetes, whether they are diabetic or not. But right now we have a glucometer. We can just easily sit and just check. Likewise, in future, what I hope that if they really come up with a efficient integrative platform, and these approaches are very simple, like all these genomics and transcriptomics, this can happen within a day. Like using the high throughput sequencing technologies, we can profile, we can create a OMIC profile within a week or so. And the integration platform is really important. We should come up with a strong pipeline which provides a reliable result. Then definitely it's feasible. The patient can be treated using the precision medicine approach.
Okay. Thank you so much for your contributions towards the academic sessions of our students community as well as the research communities. So thank you on behalf of our management and the professors, Dr. Egbert Selvin Rose, the Jani Kumar, all are there.
Thank you, sir. Thank
you. Thank you so much for yes.
So I'm very happy to be here. Thank you so much for.
Yeah. Do anybody over here have any questions more to ask? If not, we can wind up the session. Yes, thank you so much, sir, for your presence over here and accepting our invitation and being in a platform after 7 months later, I think. After 7
Yeah,
months or
I think
8
so.
months, we are seeing together. It was quite happy to see you. And thank you for lecture over here. Thank you so much.
Yes,
Thank you so much.
And especially I want to thank again for the organizer for organizing this wonderful session. Yeah, thank you so much.
Thank you, sir. Thank you for joining with us. Thank you, sir.
Thank you, sir.
Thank you.
Thank you, everybody, for joining with us. Thank you, Lord.
Thank you, Partha.
Thank you, ma'am. Thank
you, Partha.
Thank you, sir.
Thank you, Partha. Thank you, Dr. Agency, Dr. Shansha, ma'am.
Thank you, sir. Thank you, sir.
Thank you to the speaker.
So thank you.
It was really an informative session, sir.
We understand the
thank you.
Mix and all. So thank you and good luck for future research perspectives. Thank you, Doctor.
Okay, sir. Thank you so much, sir. Thank you.
Thank you, sir.
Thank you. Bye, sir.